Hey everyone, it's Ryan here, a supplemental instructor for Bio 202. I'm going to talk about positive versus negative controls. It's an important concept both inside the classroom and out, and can sometimes even appear on standardized testing like the MCAT, DAT, or GRE. This is definitely something you're going to want to have down pat. Let's start by talking about what a control is supposed to accomplish. A control is a group that is exposed to the same general conditions as the experimental group, but the control group is not exposed to whatever experimental condition you're employing, a medication for example. Usually only one condition is different as to avoid confounding variables. The goal is to have something to which you can compare your unknown experimental group in order to verify that your experimental setup worked, establish statistical significance, and quantify the degree of success or failure. We can do this two main ways: positive and negative controls. Let's start with the negative controls because they're a little bit more intuitive. If you do research, especially medical research, you've almost definitely used a negative control before. Let's say I discovered an anti-cancer compound that we'll call drug X. I want to know if drug X works, and if so, how well it works. So I take 10 mice who have the exact same type of cancer, we can do this in the lab, and divide them into two groups of 5. For one group, I give them 3 doses of drug X. For the other group, I give them 3 doses of saline, saltwater solution. The second group, the ones who receive saline, are the negative control. So what does it mean to be a negative control? Well, I know that the saline isn't going to do anything to stop the cancer. I'm looking to see a reduction of cancer, and I know that saline will not do that. Thus, because I chose a control that I know will not produce results, it is a negative control. So if that's a negative control, what does it mean to be a positive control then? Well, let's say I found out from the experiment that drug X did successfully fight cancer. Great! I've learned this because I compared it to the negative control. But I'm worried that drug X could have damaged the liver. I need some way to make sure. Luckily, I know of a compound, NAPQI, that is known to damage the liver. Fun fact, this is a real compound. It's a metabolite of Tylenol, which is why you should always be careful with your dosages. So, I take another 10 mice with cancer. I split them into 2 groups, and again, I administered drug X to one group. However, instead of administering saline to the other group, I administer NAPQI. Remember, I'm not looking at the cancer anymore, I'm looking at the liver. I know that NAPQI will damage the liver. While in the early experiment, I knew that my negative control would have no effect, in this experiment, I know that my control will have an effect: liver damage. Thus, it's a positive control because it will cause a previously known effect. I look at my results. My positive control, NAPQI, has liver damage. Therefore, I know that my experimental setup worked because my positive control turned up the expected results. My experimental group, drug X, does not have liver damage. I know this by comparing it to the NAPQI group, our positive control. So in summary, I used a negative control, saline, which had no effect, to verify that drug X worked as an anti-cancer drug. I used a positive control, NAPQI, which did have an effect, in order to make sure that drug X did not cause any liver damage. The important things to remember: a negative control produces no change. Negative and no both start with N. A positive control produces a previously known change. Positive and previously known both start with P. Let's do another example to hammer this home. I'm a doctor who has just seen patient A, who I think has strep throat. I take a swab of their throat and rub the swab on a bacteria culture plate named plate 1. If patient A has strep throat, I'll see colonies of Streptococcus pyogenes growing on the plate. If patient A does not have strep throat, I will not see any colonies. I decide to make 2 controls. Plate 2 was rubbed with a blank swab that I know does not have any bacteria. Plate 3 was rubbed with a swab that I know is contaminated with S. pyogenes. Which is the negative control? How about the positive control? Pause the video here and figure it out. Let's go over the answers. Plate 2 is a negative control. I know no bacteria will grow. There will be no effect. Plate 3 is a positive control. I know that bacteria will grow, so there's an expected, previously known effect. Shown on the screen now are the results of the culture. Does this patient have strep throat? Pause the video here and figure it out. Yes, the patient does have strep throat because their sample matches the positive control. Let's do a different problem. What if I'm testing a patient B, that I know has tuberculosis? I want to know whether or not patient B's Mycobacterium tuberculosis, the bacteria involved, are antibiotic resistant. I plate it on plate 1. On plate 2, I put antibiotic resistant tuberculosis. On plate 3, I put antibiotic sensitive tuberculosis. On all 3 plates, I administer antibiotics. Which plate is the negative control? Which plate is the positive control? Pause the video now and come up with your answers. Alright, let's go through this problem. Remember, a negative control is one that will have no effect. The effect that I'm looking for is resistance to antibiotics. Plate 3 is sensitive to the drugs, so I know that the bacteria will die. Thus, there is no resistance. There's no response. It's the negative control. Plate 2 is resistant to the drug, so it is the positive control. This will end up being important when we're looking at the Ames test. It can get confusing. If you end up confused, try defining what effect you're looking for and write down your logic. It helps. Shown on the screen now are the results of the culture. Is the patient's tuberculosis antibiotic resistant? Pause the video here and figure it out. No, the patient's tuberculosis is not antibiotic resistant because their sample matches the negative control. So in summary, when we're talking about controls, a negative control produces no change. A positive control produces a previously known change. At this point in the video, I'm going to stop talking and there'll be some practice problems. Pause at each problem, solve it, then press play and move on to the next one. The answers will be at the end, and if you have any questions, feel free to bring it up at peer mentoring or SI hours. Good luck!